Okay, if you pay attention to your channel, the week 14, which last week, next week will be your presentation. Here I just show you the score sheet, what it looked like. Very simple, 4 categories of the scoring criteria. And each of you come here, and then the other students will score your significance. Okay, for example, Zara get a very high in her significance. She will be ranked number 1, and you will be number 2. I don't know who's here. She's not here, so number 6. So you rank your peers, okay? And then at the end, I will sum the rows, and then that's your total score. Then rank higher, and you will get more points. Rank lower, you'll get less points, okay? So significance in innovation is the topic you selected to discuss whether you have some new ideas, which just like the homework 3, it's a random forest, but you use random forest algorithm classification to do the different directions to solve different problems. And the approach is you present your proposal or your preliminary study and see whether the whole process makes sense. You select the correct method, a good algorithm, and there are good rationale why you choose this, not that. And the performance is how you interact with your audience. Okay? It's fair? So I will print out some copies for this. Questions? Yes? Yes. So these presentations are next week or we haven't decided yet, so still in the stage of voting. So most likely if you'll be, I think most people vote for. If your final is 6, then we have to do 29th, the most likely to be. But prepare maybe late, okay? But I believe most likely to be the week after this. So we haven't finished voting yet, but yeah.
Does it look like your final exam date, people prefer May 6th? If May 6th wins out here, does that mean the final date gets pushed back to May 8th?
Yeah, I think so. Yeah, maybe. Yeah. Yeah, I feel I gave you too much freedom, so it's hard to choose. Yeah. Yeah. Yeah, some of the analysis. You can do it completely out of the box, but some principle you learn from the class you want to apply to them. Yeah. Yeah.
On average, should we aim for like an introduction of like the basic analysis of how we learn like the TE then TB, and then we should go for enrichment, and then we should go for different specific things we learned, like or try to learn like the module 3 and stuff like that.
Yeah.
Or
yeah. So you are talking about the presentation or homework? Presentation, yeah. Presentation, I hope you'll go beyond the homework number 4. Right. Homework number 4 is very limited, just play with a DV. But the final should be, the presentation should be expanded version of that, rather than just use DV. Yeah, so so we have spending 13 weeks to discuss from a very basic vector matrix, multiplication of matrices to today. So the point I want to make is in regular molecular biology lab, they will do single cell RNA-seq data. Okay. we can use this to measure the expression of a cell type specific gene expression. But eventually, most of them just compare to conditions, okay, wild type versus knockout. They will use DE, differential expression, to find, okay, which gene upregulated, which gene downregulated in which cell type. That's very common protocols, procedures. I think it's on most of people's mind. But the whole class of here is try to let you know there's more than that, okay? So network analysis is one of the extension, okay? So network. So your last homework is DV. We try to consider not only average difference between gene expression across two conditions, but also the variability. So that's the power given by single-cell RNA-seq data. So you cannot just average everything and compare, say, okay, this gene on average is higher than that gene on average across all the cells. But we have access to variability. That's another level. So at least you know we can just not only rely on this routine and single-cell RNA-seq to DE and enrichment analysis. At least we can do DV, we can do network analysis, okay, to learn more at systems level. So we will look at the definition of a network or graph first, then introduce some systems biology or network theory for the nodes. It's called centrality, a lot of measurements for the nodes' properties, how important these nodes in the network. Then I will mention a method called WGCNA, okay? So this is pre-single cell data time. People invented or proposed this algorithm, has been widely used for bulk RNA-seq or microarray data. I bring it up because this is so popular. If you search how many papers cite this, use this approach to do the network analysis, you will be surprised, the citation, the numbers. So we have to know this as a common knowledge. If you learned network analysis, you never heard about the WGCNA application in genetics, that's not complete. The problem is this will introduce quite a lot of false positive. You look at the two genes, you believe through the comparison of the samples, these two genes are positively correlated, and it's linked to make a network, but in reality, the power is really, really low. So just let you know, there's here. And then we will talk about the subnetwork, the community detection of modules. Then move on to single-cell gene expression data to construct gene regulatory network. And then there's a method called GE3, and it's one of the best so far. The people believe this is a random forest classification based, but they use it for regression. So that's very special. So the category classification regression tree, which is another method. And then we will introduce multi-layers networks, and then the suite network-based analysis, which you can use for the network analysis. So this is a set of the tools we developed in my own lab. So I will give you an introduction at the end. So we can have functional network data from different experiments. This is a reasonable sample from MRIs, maybe, functional MRIs. This gives you the signal from one position or one sampling point in the brain, and then here's another. The good thing about this sampling is you have time series data. So you have this cross time interval, and you have two signals. Then you can do the correlation between this curve, these two. If they are correlated, then you put high values in this correlation matrix. Then you sample some other signals. Then you have a correlation matrix. So this correlation matrix can be considered or used as an adjacency matrix. Adjacency matrix is a representation of graph. So this will be automatically form a network. Okay, so this network will tell you different regions in the brain, how they functionally linked, because you have these MRI signals. I think neuroscience people use quite a lot of those analyses. So very basic knowledge terminologies. If we talk about network, or sometimes it's called a graph, so you have a node and link, or you call vertices or edges. You can represent network using adjacency matrix. I think you know this already, because we talked about KNN network, we talked about previously, okay, right? So the network people has been using a series, many of those called centrality measurements to quantify, to give a number, say which node is more important in the network. So we need to have some idea about how those are computed. So the simplest one is called a degree. Okay, this purple node is connected with 1, 2, 3, 4. Okay, immediate neighbors, the connection. So you just count how many neighbors are connected with this node. It's called a degree centrality. For example, this one here is just 1. Degree level is lower than the purple one. So this is the most fundamental ones. Then people also propose another called closeness centrality. Let's come back to this. So this is the closeness centrality higher in here, okay? It's not too much different from a degree, but it is computed differently. So you count the distance. So let's see this using examples. So A is one of the nodes in this network, have five nodes. So you compute the distance from A to B to C to D to DE. So those are the shortest distance you can get from one node to another. So you then sum the total shortest distance from your node to the others, then use 1 over it. So this is the number which calculate as a closeness centrality and gave to the nodes. So node A has 0.1. Well, node B, if you calculate in the same way, you get 0.14. Node C have 0.16. And D is symmetric with B, and E is symmetric with A, so you have the same values for D and E with B and A. At the end, you can use the absolute value, so 0.16 is the highest. So C has the highest closeness centrality. So this is a measure of how center you are in the whole network. Because if you are in the center, your distance, your travel distance to all the other members, all the other nodes will be small. So when you have a small distance overall, then because it's 1 over it, so your value will be higher. So this is a measurement of the centrality, how much this node is in the center of the network. There are other ways, and you can calculate another distance than 1 over it. Then so 1 over it to get this harmonic means and the summarize together. So you get a slightly different flavor of the centrality measure using this equation compared to closeness centrality. So people just play with these different definitions. Well, this harmonic centrality, if you do this way, and you will get the values which will be higher in the bridging points, okay? So let's see. The harmonic centrality for this is 2.5, and this 3.8, this 2.5 in the same position. So different equations, you get different perspectives. So you consider which perspective is more important, you get different measurements. Then I want to introduce this concept. It's very interesting. So you have so you know how to compute matrix multiplication. So if you have adjacency matrix here, so this is called M. You know you can multiplicate this. This is M will be n by n, right? You know you can multiplicate itself. So if you have M by M, and then the product of this, remember how we write the matrix multiplication? So I always encourage you to write in this way, and then you get another n by n matrix. So this operation will give you the results of the power of 2, okay? So this is n by n matrix. You can do that again. So you can do n by n matrix multiplied by another m, so you get qubit, and you can do another. So because adjacency matrix is a squared matrix, so you can do multiplied by itself again and again. So take power to infinity, and then you always get the same n by n matrix, right? So this example is for this network. And then you can define this network using adjacency matrix M and using this way. So this is a directed network. So it goes from 2 to 1 is linked. So that's why here you have 1. Well, this network is not so from 1 to 2, there's no link. So that's why you don't see a 1 to 2 link here. It's 0. And so this matrix is not symmetric. This gave you the idea it's directed. Okay, so You know how to do this. It's very easy to compute, right? So now our question is, what's the geometric interpretation once you take the power 2 for this, so m times m. Let's see, if you m multiplicate by itself, you know how to compute, and then you get this, okay? So what this tells you is so what this adjacency matrix tells you is from 2 to 1, you use one step, you can reach from 1 to 3, you can have a parse from 1 to 3. So because 1 to 3, there's value. Well, when you take m times m, then you get this matrix. This matrix will tell you how many different ways you can reach from node 1 to 2 by how many different ways. Okay, so now by 2 steps. So this 2 indicate if you by 2 steps, how many different ways you can reach from one node to another. Let's see. So this place is 2. What that means is you go from 1 to 2. By 2 steps, you can go from 1 to 3, and then from 3 to 2. This is one possible parse. Another is from 1, you go to 4, and from 4, you go to 2. Okay, so this number 2 tells you from node 1 to 2, you have 2 different ways. Take 2 steps to reach it. So this is an interpretation of when you do this, the power to the adjacency matrix. Depends on how many of this power you take. It gave you the information of how many steps and how many different ways. Let's count another. So this is 3. So what does that mean here? So this 3 means from node 1, 2, 3, 4 to 1. Okay, you have 3 different ways. You can go from node 4 to 1 by 2 steps. Okay, so from 4, because this position is 4, 1, okay, to 1. The first way is go, okay, by 2 steps. The second way, right? What's the third way? Go 5 to 1. Okay, so this number tells you from which node to which node. If you use 2 steps, how many ways you can go through. So same idea. If you compute the qubit and 3, then you will get possible parse from one node to another by 3 steps. So the power of adjacency matrix tell you the numbers of works from one node to another through the steps. And the steps, number of steps gives by this power. So why it's useful? It will give you some idea the connectivity between one node to another, which not necessarily to be directly connection. Okay, if you look at these 2 nodes, maybe it's not connected by direct link, but you can move from one node to another by 2 steps, and you also want to know if I allow 3 steps indirect connection, how many possible ways I can move from one node to another. So this gives you a analytical power to detect or measure the indirect link or connections between nodes. So it's a very, very important concept. So people develop this so-called cost index by considering not only the direct link, so this is the adjacency matrix itself, this is a network, and also consider possible links with second and the third and even more. So this will consider all possible directed, undirected, and by different number of paths. So when they put them together, so you can get this matrix, and even higher power, and put them together. And this cost index will tell you not only directed link and also indirect link by many, many different paths possible. So this is called total number of works. Let's see, for the same network, if you only consider the degree, you may think, okay, those nodes are important because they have a highest degree. Well, if you use closeness centrality, so in the B, you see those nodes are more important. They are in the center. And the degree may be not the highest, because the highest degree is indicated by this red color, which the nodes are here. Then if you use harmonic centrality, and those are more important, well, depends on the cost index. And those ones are the most important. So if you consider this as a gene regulatory network or protein-protein interaction network, use different centrality measurements, you will get different views. And some protein, some gene product may be more important when you consider all the possible paths through these nodes, and then using causal index as a centrality measure. Consider the degree, say which one have more connections, direct connections. Those may be more important. So this gives you different ways to measure the importance of the nodes in the network. So normally we will consider multiple different centralities and have for each node you have 4 numbers maybe to tell you if you want to add more to emphasize some other properties, you can. For instance, this is called the leverage centrality. You have a different definition. What I found is those papers are easy to read because they are pictures. Okay, you have geological this interpretation with the graph. You have a geometric interpretation. You always see, okay, what the K is, what the IJ is, and so it's not so abstract, not like eigenvalues, those things. So any other questions do you have? Are we okay? So this is a very, very basic network theory and using centrality to measure the node's importance. Now I move to WGCNA. So before that, I would say a lot of those results may be false positive, not reliable at all. The reason is without single cell data, if you consider how the gene regulatory network can be constructed. So the idea here is we want to have gene 1, gene 2, gene 3, those ones. For example, we want to know whether this gene and this gene are positively correlated. Okay, so you want to build something. You want to see whether this is linked or this is linked, so they're all question marks. So we use correlation. So when you build a correlation, you need to have a scatter plot, right? So this is gene 1, gene 2. You need to consider what your sample is, okay? What are your samples in order to build a relationship between these two genes? You need some points in order to see, okay, these two genes correlation is positive, so that's why you can put a link between them. If there's no correlation, then you won't put any link on it. But we need to consider before the error and the error before single-cell RNA-seq become available, and how can you get the samples? So what's this point here? What is this point? So you need to compare two conditions in order to get those. Let's see. You need a condition 1, the simplest one. It will be one group of animal, and then knockout sample, for example, here, right? 2 samples. Then you need to have a gene expression value. So this is a gene 1. Well, in knockout, the gene 1 become higher. So that's your sample. You need also have a measurement of gene 2. So you have a gene 2 is here, and you hope gene 2 in this sample also high. So you need to have those relationship. You link this, in order to show you the correlation, okay? So, but this is just one sample, so this is 1 point. Two points. So if you have this, you know there's only 2 points. One is here, another right. So for the bulk RNA-seq or microarray, you can never achieve enough data points in order to establish this correlation, strong correlation. You need, even you repeat, you see, okay, I repeat this multiple times. This is 1. I do this 2. I use many animals. Then you have some kind of replicates. I don't know how to set up this, but you have a few more points. But still, you only have one of these very you control for variability across individual samples from different animals, but you still consider them give you the same signal. So eventually, you still have the same theoretical points if they are positive correlated. So even though you add a few more, so samples, 1, WT3, and here you have knockout 1, knockout 2, kill 3. But they all tend to be cancel each out in theory, and you still only have 2 points to make this correlation. It doesn't make sense at all. So if you want to build a network, say you have 200 genes or 2,000 genes, you have to repeat this between genes many, many times. You'll never be able to get enough samples, give you enough points to build those relationships. But that's the place where before the era of the single cell, people have to use this way. They want to build a gene correlation network to give some idea about the co-expression, okay? So although it's underpowered, but still at that time, we still have to use and generate lots of this unreliable interpretation of data, and use many samples, tissues, animals, repeat. But there's nothing we can do, okay? That's at that time, that's the case. So this package is called Weighted Gene Co-expression Network Analysis. It's R package. It's been developed in the UCLA by the Steve Haworth in 2008, and have been cited many times. So we start from constructing a co-expression network by using this, okay? Then manipulate somehow, build modules, and then if you have some other information, you can use this as a covariant and to regress into it, but not important here. And then from there, it start to find those modules, which is a group of genes tightly correlated to each other and form a module. So it's some sub-networks. And then they will do the enrichment analysis to see the function of the genes in each modules and whether they share some similarities and corresponding to any function groups. So that's what we start with. You have 2 samples and some genes look like these red ones, okay? Positive correlated. Green ones, negative correlated, but they all move to the same direction, okay? So when the correlation is strong, we just put a strong correlation values. So you can use Pearson correlations. So we hope there are modules, and the blue, green, red, and then there are external information to see whether they're responsible for some functions. So there are some treatment. So we can take correlation between genes and then take absolute values or become unsigned. So transfer the correlation matrix into a genesis matrix in order to build the network. You can do a transformation like this. So this will put your values into between 0 and 1. The problem was this correlation is ranged from -1 to +1. And when we build the network, we want to put values here. If there's a negative 0.75, and here is a positive 0.75, 0.6. So it's easy to deal with positive ones. And people don't know how to deal with this negative correlation. If you think about, if you have a negative correlation, means this gene and this are anti-correlated. If these are positive, so this forms a cluster. And then those ones will be negative against the whole thing because they all positive correlated go up together, and then suddenly you have another cluster move against it. It's very hard to interpret in reality. So that's why they did this transformation. So if you do this, then this correlation will translate into 0, 1, I believe, or 2. So you get non-negative values. They all deal with only positive ones. And then once finish the transformation, so we ignore the negative ones, but transform that into a so-called still the standard, but then we just deal with these non-negative values and set a cutoff. Okay, so if you have a value greater than 0.5, then I will keep this, say 0.7. I will keep this. If it's smaller than 0.7, I will get rid of it. Only keep the value which is strong enough. You can also select the soft cutoff and make it a little bit selectable. Then start to compute a so-called topological overlap matrix. It's another way to smooth out the signal from the raw correlation data. So if you look at this, it's before doing this transformation, and this is after, then you start to show all these genes share the similar topological properties, and they should be considered as a group. And this topological overlap measurements is one of the proposed steps in this algorithm and make it more successful rather than the raw data. So raw data will tell you some connections between modules, but this transformation take into consideration of the topological properties of nodes and somehow smooth out the structure, makes this more interpretable. So for example, those are not connected. It will help you to group them. There are some details in how this was computed, a little bit unnecessarily so not very useful, but the idea was borrowed from this Bababasia, who is a very famous network theory people. And the idea was borrowed from this paper, 2002 paper in the Science. So this will consider a transformation. So you have some of those details we can work out later. It considers the neighbors, okay, and also it considers the direct neighbors and And also consider the shared neighbor. And if you have two nodes, have a lot of shared neighbors, and this index will help you to merge these two I and j a little bit. So this gave you idea about how to compute those values. We don't have to spend too much time on this. You can do that at two different levels if you consider just one-step neighbors, and you get a smooth picture from this before transformation to the GT, the TO TOM1, and you can consider not only the immediate neighbors, but also two steps neighbors. If you consider I and j, and there are two-step neighbors, and see the overlap between I and j, and you will have a even smoother this transformation. So if you look at the picture, and this gives you a smoother, more connected neighbors, because they merged those with large overlaps between topologies. And you can do the 3 steps, okay? This is another comparison. So once you use the transformation, and there's another version of this, and you can start to merge this very sparse modularities. But now you merge them because you use the topology, the shared neighbors, and merge those nodes, and then you start to see these large communities. So this is just one and another one. So you can, after you do this topological transformation, then you can easily detect the modules. And each module may contain hundreds of genes. You will see a lot of those cluster dendrographs in the paper, and each of those colors indicate a module, and they will do the enrichment analysis, see all the genes in the same module, and the green module will have gene associated with apoptosis, and pink-red color module will associate with a cell cycle, something like that, okay? So you found the analysis look like that. Okay, so the whole idea is the information from WGCNA is from a bulk RNA-seq or microarray analysis generated data. So they're limited by the number of samples. You force the correlation coefficient, no matter how you transform, utilize all this smoothing technique, eventually you will get some unreliable data. So I wouldn't recommend you reading those papers, because the principle are not hold, because you never get enough samples to really find the relationship between genes. Okay, so we move to this short introduction of the modularity or detection of modules in the network. Once you construct a huge network, if you use a very relaxed cutoff, so most of the nodes will be linked, you have the huge spaghetti looking at the hairball, and it's hard to interpret. Everything is connected with others. So people use some technique to find those highly connected sub-modules, and within this module, the nodes are highly connected to each other, but the between modules. For example, this one versus this, you have to go through another bridging nodes, so they are not highly connected. People want to find those modules, see whether there's structures in your huge global network. There are major structures within there. So the most commonly used method is called the Louvain algorithm. In Surat package, people build this cell-cell network and then use Louvain to do the classification in order to see, okay, one cluster is one subtype of cells. In my toolbox, I didn't use this, but in theory, I can. What I use is t-SNE dimensionality reduction, then use k-means to overcluster cells into small groups, then merge them, depends on the annotation. In Surat package, they did the dimensionality reduction to the 50 dimensions and use KN, I build a network, and then use Louvain to find these subnetworks. Each subnetwork assumed to be the same group of cells. So they annotate submodules in that networks and gave the name of the cell type. Okay, so that's where this algorithm is applied. So this network this algorithm contains optimization problem in order to achieve this, make this queue, I believe, this as small as possible. So this queue contains the information, the weight between nodes. Okay, I need to. If these two nodes are in the same module, then you will compute differently if these two nodes are in the different modules. So you have to optimize the membership of each module to achieve all the within the module, those distance are on average small, as all the members is highly connected. Where it's out of the module, then you give a high distance between them. So this function eventually converged and gave you the membership of each node. If these two nodes belongs to the same module, they will have the same membership ID. Okay, there are programs to compare so differential co-expression gene modules. So those are not very commonly used, but there are possibilities that you can find across two different conditions, whether the co-expression network you detect shift or changed, you can see what's module changed. Okay, now we move to the single-cell RNA-seq data. Now we are giving enough samples, right? So in the wild type, we can have thousands of genes, thousands of cells, okay? So in this case, the cells from a wild type, you can have, say, 2,000. And from knockout sample, you can have 3,000. So easily, you get enough samples to do this, right? And also, even within the wild type, you have cells in different stage. Remember the pseudo time? Even the wild type, you can build the trajectory and the cells in one stage translates to another. So this gave you the perturbation, internal perturbation. So you know the small difference and introduce, not like bulk RNA-seq, you assume all the animals, give you a tissue or samples, you just want to average them, so to control the variability across individuals. But in the single cell conditions, you have the internal variability, the gene expression change a little bit between one cell to another. So those variabilities give you the power to detect whether these two genes will change together. So I only trust the single cell data constructed network. So, so this idea is to utilize this matrix to work out the gene interactions. So this is an example. Have I shown you this before? So this is real data, okay? It's just express spreadsheet. What we can do, just do pairwise comparison. So you can find one gene against another, one gene against another, any two of them. So those are the raw data. Remember, you can do magic imputation. Once you impute the magic, using magic, this zeros will be imputed with some numbers, okay? That will help you to do this regression or correlation, because if you have only zeros, then you only have these points all located here, will not very powerful when you do the correlation related. So in this case, we just do the pairwise regression or correlation between genes. For instance, this is two genes from a sample from lymphoblastoid cell lines. Each point here is a cell. Okay, we can easily get thousands of cells from single cell data. This has been imputed with magic, so you can see there are non-zeros, and then non-negative should be. Oh, there are some negative. This imputation result in those. But they are not located, although this gene may have a lot of zeros, especially for these highly variable genes. A lot of gene just not expressed in this cell, but suddenly expressed in another cell. So imputation help you to find the correlation between these two genes in those cell population. Well, now we keep this gene the same, AICDA. We'll change REL to ARF4. And now you can see the results. This looks like nonlinear relationship between these two genes. So when you do the correlation, then you may get something small or close to zero because there's no relationship. Although looks like there are some correlations. But now if you look at the third gene, remember each point is a cell. So if you look at the third gene, the PRDM1, all these genes are in the NF-kappa B signaling pathway. It's very important for the immuno signaling. So if you look at the third gene, so this gene, when the cell expresses this gene high, we color them more yellow. If this gene expression level low, we put the color into blue. So if you look at these clusters, the whole thing here, those lymphoblastoid cells have low expression of PRDM1. Then in that case, these two genes have a negative correlation among the cell population. Well, when the cells have high PRDM1, which relatively high, which in those groups, that these two genes show positive correlation. So because of the number of cells and the cell in the different stage, so as I said, although this is a cell lines, it's supposed to be highly homogeneous, but still you start to see this functional perturbation or different stage of the cell. So it gave you the power to build a network, and because the number of cells is high, so you have the power to delinearize the relationship between multiple genes, right? You'll never be able to achieve this using bulk RNA-seq. You can never get enough samples to show this. So as I said, this is an NF-kappa B regulatory network module, which is a working model based on B cell activation, and the connections has been worked out by cellular biologists, immunologists, using cell line experiments, using animal models. So all these solid lines are not from the data, not from single cell data, but from the previous experiments over the many, many years accumulation of knowledge. So people know the relationship between those genes. As I said, we have PRDM1, ARF, and also this ARCD. They're all the key members of those regulatory networks. If you look at this picture, if we only have single cell data and look at those genes, we can reconstruct almost the whole subnetwork here. Okay, all the solid lines are confirmed by single cell correlation. So if you look at the correlation between these two genes, a positive BRCA2, mostly can be confirmed, and also consistent with the experiments data. So it's consistent from previous knowledge. Some of those relationships are not confirmed, so it's only you can see those from the single cell data. So the idea is we can, if you have completely unknown network like this, you have never worked on this, we have no animal or cell line or cellular experiment before, we have nothing, but only use single cell data, we almost can infer the relationship between those genes. I would say 80%, okay, because all these solid lines have been confirmed with the relationship between single cell data. So that's the power. You can use network inference to infer gene regulatory network and also cell type specific. In this review paper, say, in bulk RNA-seq, you put everything together and you build a network, not reliable, but here we can separate different cell types and build a network for different cell types. And each cell type, if you have enough cells, you build a high resolution network. So I mentioned this correlation, okay? So this, I want to very quickly show you, so this is a correlation we used. So before the break, I want to quickly show you, there's another correlation coefficient. So this method is called Chatterjee, okay, the author's name, Chatterjee correlation coefficient. Normally we use Spearman, Pearson, candle, okay, so they are commonly used. But in 2019, not a long time ago, Chatterjee and found a new way to define a correlation coefficient. An example is like this. If you look at the Spearman correlation, for this correlation, this is a 0.9, and the Chatterjee is 0.89, okay? So similar. And then in this case, traditional Spearman is 0.89, and then Chatterjee is 0.57. Well, in this case, it's negative. Chatterjee is 0.87, and in this case, there's almost no correlation. So Spearman is 0.3, Chatterjee is 0.16. So the absolute value for Chatterjee is always corresponds very well with the traditional Spearman or others. However, when you deal with this kind of case, and the traditional correlation will give you 0.01 because there's no correlation between X and Y. Well, Chatterjee, this new coefficient will give you 0.76. In this case, Chatterjee gives you 0.5 and nothing from the traditional correlation. This case, Chatterjee gave you 0. Can you see? I don't see. Can you see it? Okay. Yeah, Chatterjee gave you 0.81. While traditional correlation gave you nothing. This case, 0.4, nothing. So if you have relationship between two variables, so Chatterjee, this new correlation coefficient will bring you some new perspective recently, okay? So, so my students, one of my students is working on this, see whether we can apply this new correlation coefficient method to single cell data, especially when the conditions look like this, okay? All right, let's take a 5 minutes break. innovation score, then you design a new project. If you just repeat, your supervisor gave to you, the other students will feel, okay, the innovation is not so high. I remember one of the previous students, he proposed, he wanted to detect mRNA, microRNA information from a single cell data. He even proposed to change the protocol of text genomics a little bit by adding something so very, very unique. So he got a very high score. And yeah. So think something cool and yeah. Yeah, maybe you can use, I don't know, network analysis in something which people have never done. Yeah, you can. Well, you use other people's data, the people generate this using R and something else. Yeah, as long as you can support your point, yeah, doesn't matter. Yeah.
So it's more like if you're taking somebody else's data and like doing a different type of analysis than they did to uncover maybe a different question.
Yeah, so you can see, okay, they didn't find this at all, but we have new insights from using their data. Or you found a completely opposite conclusion from that will be highly innovative, right? Yeah. What's that? If you identify the gap of knowledge, nobody have ever done this, and you found something, combine the different data or using a new way to analyze it. So just open your mind and try to be creative. Yeah, so my whole idea is I treat you as my graduate students in my lab. So if you contribute a good idea, that's the most important. Okay, so advantage. So we have large number, so thousands of cells. And also it creates automatically nature time theory. Okay, so this you can also collect at different time points. So this make it even strong. So you have a change of the cells and the two genes co-expressed at different time points. So this is a very strong indication. So there's a lot of advantage of this. So this is a review paper a couple years ago. See, we have several different algorithms we can use to build gene regulatory network using single cell data. You can have a trajectory analysis in the middle, so give you some pseudo time theory information. You can use that into your building. Some of those programs using that pseudo time, for example, this SC code. You can compare, okay, after you build a network, you can start to compare so-called differential network analysis. Everything is a cell type specific. You can compute certainly just one whole network, static. So this is illustration of using pseudo time to get this dynamic of the network change. So you can follow the time, and for different cells, build a different network, and you probably can work out this dynamic change. It's very difficult. So those are the some of these methods of the gene regulatory network construction using single cell data, which have been reviewed or benchmarked. So this is called a B-line study, and they prepared some of those small network including 20 genes or 25 genes, and they know the ground truth, so-called ground truth. They have rigorous test biologically, say, okay, this gene should connect with that gene, and they should not connect with those genes. So they prepared this small network and gave to all different kinds of algorithms, see which one perform the best. So those are the methods they tested. The different features and the ROC curve and the precision and the Jaccard index, so different benchmarking criteria they use. Those are the input network. So this GSD is the largest, the network for the testing with most of the genes in there. So this is so-called ground truth. And then they test this one, and that one for different algorithms. So the problem with this B-line, so this B-line evaluation framework is the largest network is too small. So this GSD network only consider, I count how many, some 25 or less genes. So it's not a very good benchmarking tool for more complex. If you have 2,000 genes, you want to construct a network, you don't have any good reference ground truth to test your algorithm. And this so-called ground truth, the network is too small. So when you have a small network and a lot of algorithm may overshoot, or some algorithm very slow, take hours, doesn't matter. You can get a high quality network for this small toy example. But in the real application, you don't want to run that program for your 2,000 gene network, take 2 days, just not applicable. So a lot of issues with those testing as well. So at the end, we believe Gene 3 is one of the best from the test overall. So Gene 3 is based on, Gene 3 even before applied to single cell data, they have been tested in other so-called competition, Dream4, Dream5 network inference challenge. So that's activities, and there's organizer to prepare the data set. They know the results, and give this data set to a lot of programs and see your competitors and which one will get the best results. So Gene 3 has been a winner for Dream4, Dream5 competitions. So it developed in 2009, and so it's before the single cell data become available. So this method is called tree-based. It will build the decision trees, and basically it gave you, let's see. So people use the decision trees. You can use decision trees to build classification or regression to solve those problems. So this is a book about those so-called card algorithm. You can do classification or regression. So this is the idea. To do classification, you know this decision tree and the yes or no answer that give you the separation of your samples. And then you have another level of this yes or no answer, and then to further classify your samples. Depends on those parameters, depends on those cutoffs and the parameters, the measurements. So you have weight and height. So if you consider this as a gene 1, this as a gene 2, you will be able to separate the samples, okay? So this is like 2D, 3D visualization of this decision tree. Each of these levels, and you have see different levels, that's a decision tree can break down your samples into different levels. So you have here, maybe have more levels. This all depends on this binary tree separation, and using cutoff depends on the variables you put in. So when you have just several levels, then this becomes a classification problem, and you group the sample into different stairs here, okay? But if you give you enough levels, and the level become so continuous, then the problem with classification becomes regression. But the whole idea is just split your point, your samples, depends on the cutoff of the variables. So there are different algorithms in stopping. So this is like another ways to show you. Looks like this only category sample points into those windows or stairs. But if you have enough this separation, you can do the regression for those points. So Genie3 is based on this tree, decision tree models. It started by give you this expression matrix first. Then I think they do this what they call this different ensembles. Let's see. So they build this multiple trees and put the genes in together. I don't know exactly. Then they combine those ranks of the contribution of genes into each other. Then they get the results. Let's see what LS stands for. I think they call the learning samples. So idea is you look at one genes and use other genes as a variable to explain the gene, which your focal gene in the sample. Then build a regression using the decision tree approach, using all the other genes expression to explain your target genes expression. And then those genes give you the highest prediction power will be genes strongly linked with this gene. And then you do this by take a turn, then you change your target gene and use all the rest gene to predict this target genes Then you change another target gene, use all the others to predict. At the end, you get this many those decision trees, and then you combine them together to build the network between genes. So you can think this algorithm will be slow. It's really slower than do this correlation. Correlation will be just pairwise, one gene against another. But here, one gene has to against all the rest to build a tree, so it take much longer than others. So there are other algorithms which utilize gene 3 as a core algorithm in the middle, and then build other information around it. For example, this is called a scenic, also is highly cited. Scenic, instead of a algorithm, is just engineering the package of the method. The algorithm of scenic is based on the gene 3. Gene 3 will build the tree from single cell data, and then the scenic also utilize the information between transcription factor and the targets from the database. So the algorithm, this method use gene 3 build a network between all the genes, and then they will only consider the link between transcription factor to their target, ignore other links, so make their results easy to interpret, because everything you see out of this scenic package is the link between transcription factors to its target and well known. So the only problem with this framework is it's not be able to detect a new link between regulator and its targets, but only rely on the database. You have this TF two targets database, okay, to build this so-called regulon, so the regulation relationship between genes. So step one, this framework is to use the gene 3 to build a network. So gene 3 is too slow, so there are improved algorithm called GR and boost. So there are some of this machine learning or data science new implementation using this enhanced version of the optimization procedures to make this slightly faster, okay, utilize the parallel computing. Then scenic, the workflow will trim off those unsupported transcription factor to the, so the trim off, for example, transcription factor not known to regulate gene number 5 and 6, although the network gave you the link between them. But scenic will only consider this known relationship and consider those co-expression indirected effects, okay? Then to do another steps to do this activation matrix. So another way is to give the score of this into the cell. So those cells will have this regulon activated among those cells and not in these cells in the purple, in the gray. Okay. So they give you some examples application of this to the mouse brain data, and they can find regulation relationship between transcription factors and among cell populations. Okay, those columns are the cells. Okay, this group cell have those regulon activation higher than rest of them. So you can start to see the clusters, and those are the regulators, key regulators, which can drive the activity of the transcription program in those cells. They can also look at the binding sites, or the shared binding sites for those location, these motifs, to define the cluster of cells. Another program is called the PIDC. It's called the partial information decomposition. So this is using mutual information. So this is a information theory based. The author claimed this is also very slow, so they use Julia language to implement the results, make this program faster, and they can achieve those. So this is another explanation of what's going on inside, and those information, mutual information or partial information equations. So this is our package called S code, and this utilizes pseudo-time information to build in and to infer this regulatory network with this information. And there are some of this differential equation get involved. This is also a method which utilizes some of the pseudo-time. Okay, so you use time delay information. So assume you have a network which you know this network is happened after previous time points. So you have some information between gene expression in this network compared to that. So they use that information, build in, and they got those. They claim this can learn the causality. So normally we don't know the direction, which one is driving which. But because you have this time delay information, you probably can build this causality information. This gene expression get upregulated and cause next time, and which gene get upregulated. So very attractive method. Now we move to see. This is actively, people are still working on these very hard problems. If you have networks you take from a different time and then form a multi-layer network, so see what kind of algorithm we can have. Though this one, you can look at the modularity change along the time, and you have the emerging of the modularity in one time point may disappeared, this module disappeared at the second point, maybe re-emerge in the third. So there are many possibilities. If you combine this modularity detection with the time theory, people want to learn this change of the rewiring of the regulations. So this another the so-called likelihood function or loss function in order to detect the change of the modules across time. We are interested in this so-called network switching. Okay, so you have the links, the switch, the membership of this node, this gene, was in this module at time window 1, but it changed to another module, playing a role there, and then switch back to a different. So there's some interesting some multi-layer analysis. If you have those worked out, if you have a real biological explanation, that will be really, really cool. So in general, this time varying graph, the algorithm is still developing. We don't know, we don't have a very solid solution, say which one you should use will give you the better results. And also the research in this time varying graph should not be only in single cell world, it should be in many, many other applications. If you found some progress in other domain of the research, you may borrow that, apply to here. So your project could be, oh, I look at the physical review A journal, and there's one guy in the world developed this time varying graph theory, and I will borrow this idea and utilize in our single cell data, and I will construct a pseudo time data, and then construct the pseudo time and group my cells into different time points, and I build multiple networks. I probably can use those information, new algorithm developed to try out. So that will be one of the examples. You ask for examples of the projects. That could be one. So people are still working on different ways to extract the information out of this time series networks. So this is utilize NMF to do this, to learn the modules, which in this case are the modules with conserved. So across all the time points, there one of these modules, for example, never change. So they want to work out, okay, what genes are get involved in those modules. So I will show you a couple of what we did in the network analysis applied to single cell data. So the first tool we developed is called the single cell 24-net. This is a tool for construct gene regulatory network and also compare. If you have two networks, how are you going to compare and get the results? So the idea is if you have two conditions, okay, again, well-known example will be wild type against knockout, or before treatment, after treatment. Then you have two gene regulatory network for the same group of cells. And we want to compare network constructed under condition 1 against network constructed under condition 2. In this case, we want to compare nodes by nodes and found, for example, node number 2 changed its regulation relationship in the two networks. So it was strongly connected with gene 6, 5, and 4, but in condition 2, the link or regulation relationship become weaker between 2 and 6, 2 and 4, but it becomes strong between gene 1, 2, okay? So we want to find those genes which show major changes in their regulation relationship with other genes. So this is one of the options which I said you should consider analysis which beyond the differential expression. So in this case, we are not compare gene to their expression average in condition 1 against its expression in condition 2. We don't care. We just care about whether the regulation relationship of gene number 2 changed in these two networks, okay? It does not necessarily change the expression average but the relationship change is what we want to detect. So these pipelines look like this. We need 2 inputs, 2 single-cell RNA-seq matrices. So this is sample 1 indicated with a black and the sample 2 in red. They are not the same group of cells, but you can see align the genes, the same set of genes. And then you can build you can build a gene regulatory network. So you build this, you build one network, and this build another network. And then to compare 2 networks. But we decided to do a subsampling because at that time, people always argue, you have single cell data, it's a sparse matrix, and also there may have unknown structure in there, so we do subsampling. See, we sample out of 2,000 cells, each time subsample 500 cells, and do this random subsample 10 times, and you construct a small subgroup of cells, a same set of genes, and then you can deal with the subsamples of cells independently, then take average. So you control for the unknown variability among cell populations. So we can do the same thing, just take some time. So see, I re-subsample cells into T subsamples for each condition. And for each subsampled cells, we use PC regression, principal component regression, to build gene regulatory network. Okay, so PC regression, similar like PCA. So if you have a matrix, you look at one gene, and then the rest of them is a matrix, right? So you do PCA to learn the major components from the rest of the genes, and use the major components to regress against the target gene, and then look at all the other genes' relationship with that gene, how the other genes can be used to explain that gene. Then you move to another gene, and again use the rest of the gene as a matrix to predict this target gene. So you do this iteration loop through all the genes. You learn the relationship using regression model for the target gene with other genes. So similar idea as I just described with the Gene 3. Gene 3 use decision tree to build the regression. But here we just use regression through the principal components to learn the major components to do the regression. So this is much faster. So we can build a gene regulatory network for each of the subsampled cells. So here we have T subsample cells, then you have T networks. Okay, each one is n by n, depending on how many genes you have. So this forms a multi-layer network. And each network should be very similar to each other, okay, because we subsampled from same sample. And then you have another multi-layer network. We treat those multi-layer network as a tensor to learn the major components, then reconstruct a network and take average. So this becomes a denoised version of network. You can construct it directly from there without those intermediate steps. But those intermediate steps will help you to get rid of a lot of noise. These two networks we will compare and using manifold alignment to assume they come from the same manifold structure. We try to align them, and then the place where this contain the same set of genes. So one gene will have 2 points in the aligned space. One is from sample 1, another one from sample 2. If they are well aligned, these two gene, 2 copies of the same gene will be very close to each other because they are aligned very well. If this gene changed the distance a lot, so we will consider this gene will be the so-called differential regulated genes. Those genes change their network relationship with other genes between these 2 samples. So we can find those genes and study those top genes and to explain the difference between 2 samples. So this is an application. So this is neurons from a mouse, and the blue is before the control, before treatment. And the red ones are after injection, this morphine stimulations. And when you compare the average of those key genes, you don't see any difference. And also all the cells in the t-SNE plot, they're all the same. So all the neurons, there's no separation. So morphine looks like there's no impact on the key genes, which are supposed to be changed, okay? But if you build the network, if you compare this, you put the blue gene, blue cells here, red cells here, you compare network, you will find those genes, okay? The most, change the most. And those genes, if you do the enrichment analysis, you will find those pathways. And those pathways we know is true because they are morphine, we know the perturbation is morphine, okay? And so this is not a random result. This is definitely, we recovered the gene regulatory change, not the average change. Gene expression has not changed yet. So this regulation expression difference, those key genes have been detected. So another tool is very similar to the one I just described, but this time application is so-called a virtual knockout. So we simulate a gene. Once it's been knocked out, what will happen? So the pipeline is exactly the same as a 24-net to compare these 2 networks. In this case, we are not using the second sample. This is just one sample. For example, one sample from wild type, only one. So we copy this matrix. You copy this matrix, give the algorithm. It will give you the results, some random genes, okay? Because you gave the same example, same inputs, the output has nothing, just random genes. But what we simulate here is just remove one gene by setting this gene's expression into 0, okay? To simulate this gene has been knocked out from the sample. So the only change is one of the rows of this matrix. If you are going to knock out gene 1, then you put all zeros in that row to replace its real expression. So now these 2 matrices are different. They are different matrices. And then the output will tell you what's the other gene change after you knock out that gene. Okay? So we tested this using the real data, and this is a knockout. This is wild type, and this is a TRIM2 gene knockout. And then from wild type, we get the one sample example, wild type information. And then without using the information from the knockout animal, and just do the prediction, you can get all these pathways, all these genes, the top pathway changes. We get the same results as the real animal experiments. So the idea here is you can utilize this virtual knockout even without doing the real knockout, to predict what will happen after you remove that gene. So you remove TRIM2 of the other genes that will change, you can get the results. So this is very useful. You can knock out any genes you want. You can do the double knockout. You can do triple knockout. And sometimes double knockout will kill animals, so you'll never be able to do that. Some housekeeping genes, you will never be able to do that, okay? So network, okay. I think I already mentioned this. Interaction is also based on the network. And you build a network for cell type 1A and cell type B and their corresponding network and to learn. So the framework is rely on the construction of a gene regulatory network. For details, we have publications. You can take a look. Yeah, I think that's all for this semester, the lectures. And Thursday, we will play with all these network construction tools. And I can review the DEDV or the pipelines to finalize your learning. Okay? All right.